Good morning. I'm Jacob Kelly, one of the Duke Cardiology Fellows at the DCRI, here with Dr. John Alexander to discuss, who just talked about late-breaking clinical trial Regulate PCI by Dr. Roxana Mehran. Dr. Alexander, what are your primary takeaways from this presentation?
Yeah, so Regulate PCI was a large international clinical trial studying Reg1, this new anticoagulant, versus bivalirudin. And the trial was actually stopped early because of some unexpected safety concerns related to allergic events that occurred more frequently in the Reg1 arm than in the bivalirudin arm. So we didn't end up with the 10,000 plus patients in the trial that we expected. We ended up with about 3,000 patients in the trial before it was stopped. And with those 3,000 patients, Reg1 had no benefit over bivalirudin on ischemic events, so really similar rates of ischemic events in the two arms. And there was a numerical excess in bleeding with Reg1 compared to bivalirudin. There was a statistically significant increase in minor bleeding, but a numerical excess in more severe bleeding. So overall, not a positive trial. Similar ischemic events, more bleeding, and then this very interesting allergic reaction response, which maybe we'll talk some more about.
That's interesting. So you discussed the ischemic events, you discussed the bleeding events. Tell us what it was like working with Regatta, which is kind of an interesting company and a really interesting backdrop with Duke.
Yeah, so this technology, Reg1, it's an RNA aptamer factor IX inhibitor and a reversal agent. They were actually discovered in a Duke Department of Surgery laboratory a number of years ago by an investigator named Bruce Sullinger. They spun out a company called Regatta. Bruce is still at Duke, but this small company, Regatta Biosciences, was created. And then they took this small company took this drug up to into a phase 3 clinical trial. They raised several $100 million to conduct a very large clinical trial, and they were a great collaborator. In a small company, you have a much smaller group of people you're working with. And my colleagues, Roxana Mehran, who presented the results, Mike Linkoff from the Cleveland Clinic, and I had a great collaborative relationship with Regatta, designing and then running this trial.
So even though this was a negative trial, what was your learning experience working with a smaller company, maybe from the ground up? Can we take advantage of this in the future?
Yeah, I mean, I think there are a lot of lessons to be learned about efficiencies in conducting clinical trials that we had the opportunity to explore in this trial with risk-based monitoring and designing quality in from the beginning, working collaboratively with the FDA on that. And then I think this whole idea of reversible anticoagulants or reversible therapeutics is also a novel platform. We had hoped that using a high dose of a factor IX inhibitor with reversal, that we would get efficacy, maybe more efficacy than we see with bivalirudin, which has some residual stent thrombosis signal. That with this new drug, we'd get better efficacy, and then with reversal, be able to prevent bleeding. And we didn't see that, at least not in the 3,000 patients or so that we studied. There's some lessons learned. We only reversed the drug 80% of the way. Maybe if we had reversed it 100%, we would have seen some different bleeding signal. It's unlikely that would have affected the efficacy piece. And then the other really interesting piece from this, from a science standpoint, is these allergic reactions. So Reg1 is an RNA aptamer that's coated with a PEG, polyethylene glycol PEG, to give it a long half-life. And there's some suggestion that that PEG is stimulating allergic reactions in a subset of patients. And we have a lot of blood samples from the trial and a lot of interesting data collected that we're going to learn more from about these allergic reactions. And it may move other fields of science forward as well. So these trials, even if they're not positive for the primary outcome, you have an opportunity to learn a lot from them, either related to what you're studying or ancillary fields. And I think that will be the case with Regulate as well.
Well, great, Dr. Alexander. Thank you so much for joining us today. I think the primary results are very interesting. It sounds like this has moved science forward and trialing new medicines and new science. For further scientific discovery. Please follow the Duke Fellows blog throughout the ACC scientific sessions. Thank you.